Good morning, everyone. Let's throw the room into darkness. Hey, so we've been covering some of the different tools by which we can either reduce the bacterial load or remove bacteria. Just a little point. Very often at ADM and Tate & Lyle, when they run big fermentation processes, they've got tanks that they will grow lots of bacteria. If you ever look on your candies and foods and you see something called xanthan gum, that is a product produced by a bacterium called Pseudomonas xanthii. It's a goo, kind of like sort of a pudding-y type stuff. It's a thickener. Anyway, they are doing all kinds of fermentation all the time with bacteria and yeast, and they feed in products from corn, what they call corn steep. And that would be stuff that maybe is left over after high fructose corn syrup production. Sometimes they just grind up the corn and throw it in, and all kinds of different things can be done. Well, when they are done with an experiment, they will go ahead and siphon off the bacteria into what they call a kill tank, which is something that can really crank the heat up. And they can run the heat up to 180, 200 degrees Fahrenheit and hold it there for a certain amount of time. And then they are allowed to either dump it down the sewer or they use it for various things. But it's dead. The point being, what they do is there is a health safety officer that is checking that fluid to see just how dead is it. And it's not like it is completely dead. There is still bacteria in it. They look for something they refer to as a 6 order of magnitude reduction in bacteria, which is, a 1 followed by 5 zeros. So, what is that, 1,000,000 times reduction? It's not 0. It is, though, a really big reduction in the bacterial load. Because getting stuff to be completely and totally sterile is a hard thing. And it brings up this point, is that sterile don't mean clean. Sterile means nothing living. And for the most part, I'm good with that. But there are things in your food that even though it may be sterile, it can still make you sick. I mean, we talked about lipopolysaccharide, and there are a variety of bacterial toxins that will block the salt transporters in your small intestine, in your large intestine. You block salt uptake, and it gives you massive diarrhea. And so even though the food's been cooked, if it was contaminated sufficiently, it can still make you sick. That said, if it's that contaminated, it was pretty damn nasty when it was raw, and it's probably all that fairly nasty when it's cooked. My general philosophy is, if it didn't make you sick cooked, it probably wouldn't have made you sick raw, but don't quote me on that. You hear? Don't say that. All right, so we went through then the basics of heating. That works really well. You can either heat to kill, like steam sterilizing, or you can heat to get this 6-fold, reduction in the bacterial load. Pasteurization. And then what pasteurization is doing is it's dramatically decreasing the bacterial load. Going to be have a lot longer shelf life, but it's not completely sterile. Other things we can do are things like filtration, and this actually works fairly well, where we have membrane filters, and they're a little crispy, looks like a piece of paper, but they actually are very, very crisp and very shiny nitrocellulose filters, and they can be made with different pore sizes. And they're designed in such a way that bacteria, which comparatively are pretty large compared to like a water molecule, you can pull them through. You generally have to use a vacuum. They're fairly expensive. And what you got then is the membrane filter here. You take the sample. There are going to be things that you can't heat sterilize. Things like tissue culture media that has lots of protein and stuff in it, that if you sterilize it by heat, you'll denature all the proteins and it won't work. And so you have to filter sterilize it. And filter sterilization works really quite well. If stuff is really thick, really viscous and gooey, it won't pull through the filter very well. If it's got a lot of crap in it that's insoluble, it'll clog the filter. So you have to have, reasonably clean. Everything's got to be in solution for this to work. And one of the interesting things, I don't know, has anybody ever done any camping where you've got a water filter purifier? And I haven't done this kind of camping in a while where, like, either a lake or a river, very often they're contaminated by a eukaryotic gut pathogen, a parasite called Giardia. And so you have a water filtration device that has some kind of rinsable filter system. And what you do is you have like a sipper hose and a pump, and you pull from the potentially contaminated water into a clean vessel. Now, if you're following really sterile technique, you have to recognize that anything that's on this side of the filter is potentially contaminated. Anything on this side is the sterile water, but like the transitions, like the threads on your water bottle, need to be rinsed. The cap needs to be rinsed with sterile water. Everything needs to be sterilized and cleaned, otherwise you still risk contamination. And so this same thing happens with sterile filters, is that you're putting this in, what's coming out is sterile. As soon as you open this up, you're opening up the atmosphere, and so you have to do this probably in a sterile hood, and you have to be thoughtful about all the downstream kind of monkeying around. They generally have a little plug here that you can pull out that allows you to have a pour spout, but then you got to cap it. And so if you're not careful, you can get recontaminated. A main point, though, is viruses are way smaller than bacteria. If we were to think of a bacteria being the size of a school bus, a virus is probably going to be the size of a football. So there's a big difference between a virus and a bacteria. And you can't really get a filtration system small enough that it will remove viruses. Bacteria, no problem, but viruses are really hard to remove. Consequently, our machine here, sorry, it's mostly street theater. It's not very good at pulling viruses out. HEPA filters, no, don't work for them for viruses. But bacteria, yeah, it'll definitely work. So, that's another sort of physical system. So for liquids, we've got the filter and a vacuum pump, and then you got the sterilized liquid. Still potentially has viruses in it. That'd be bad news. Gases, air, we can use a high efficiency particle air filter, or a HEPA filter, and that's what we got here. You got a pre-filter that you're looking at, and then deeper in is the HEPA filter, which is kind of expensive, so you want to have a pre-filter for that. The sterile hoods we have upstairs, they use HEPA filters. Again, what you got to do, though, is flick them on, let them run for a while, and this is something that's called a laminar flow hood, where below you, at your feet, there is an entrance, and so air is coming in, and then it goes through the HEPA filter, and then you have a fan blowing it out at you in this cabinet, and then there's a glass shield that comes down. And so you turn it on, and the air is being pulled out, cycle through, and you got this flow. And you let it run for 10 or 15 minutes to kind of pre-clean everything. We even have UV lights in them as well. You can kick that on, which will kill viruses and bacteria for a while. But you let them run for a bit, and then when you're working in the hood, you want to make sure you don't have your hands behind your sample. You always want to make sure you have your hands in front of the sample. And that way, the sterile air is blowing across the sample, across you, and then all the stuff that's on you is coming back at you. If you work behind it, you can end up having stuff blown onto you. And it's a bit of a skill to work well in a sterile hood and keep your stuff sterile. It's a test you take every day when you're sterilely transferring stuff, and you know whether you pass or not in a day or so, because if you have bacteria contamination, you didn't pass. And you generally use like alcohol, and ideally like some way to flame sterilize the alcohol. And it's a practice, and it's actually a fun skill to get good at, just because every day you get to see how well did you do the day before. And so it is possible to work within a sterile environment. All right, so those are our primary physical mechanisms. The last one, I guess what we're doing is thinking about physical mechanisms as opposed to chemical mechanisms. the physical mechanism last, and pretty much this gets everybody, is different kinds of ionizing radiation. And so we've got a variety of different kinds of radiation forms, and things like X-rays, gamma rays, UV light, and a couple of differences here. First off, most of these things are either breaking DNA, shattering your DNA. Things like gamma and X-rays, they can do that for sure. UV and gamma and X-ray as well, their primary mechanism is they're splitting water and generating a oxygen radical that is extremely corrosive, and that begins to sort of corrode and oxidize parts of your cell. So when you're thinking about radiation, you can have a direct hit that'll shatter your DNA, but more likely you're having water being split and generating oxygen radicals. So you can kind of get around this by taking a big dose of vitamin C. Taking a direct hit, though, that shatters your DNA, that's just bad news. So a large dose of radiation will pretty much shatter the DNA of any living organisms in there. And with broken DNA, ain't nobody can survive. You can't carry out cell division if your chromosomes have been broken up and shattered into little pieces. You pretty much, there's cell surveillance systems in all different cells that'll cause them to go into what's called cell-directed suicide, a process known as apoptosis. And that's actually a really cool thing for like cancer treatment. If you can shatter the DNA, even though a cancer cell is like ignoring all the rules, even they can't continue on and will continue going to apoptosis. So radiation therapy works. Now, a couple of things about this. Depending on the radiation, in fact, UV, X-ray, gamma, these are all parts of the electromagnetic spectrum, not to get too wonky. Basically, they're forms of light. And just like any kind of light here, different things can block the light. Now, UV light, it can make its way through the skin a bit, but it's blocked by opaque surfaces, certainly. And so UV light doesn't have much penetration power. Gamma and X-rays, as you know from going to the doctor's office, X-ray will go through you pretty well. They do a better job of going through things. But still, if it's thick enough, you still can't get into it. And so we can use this on different kinds of samples that can withstand this kind of oxidation and irradiation. You have to, with UV light, you got to hold it on there for a while. And it's primarily going to be used on surfaces. It doesn't get deep into things like you have a package with material inside of it. It may not get in. But that's why I'm always wondering about these boxes of milk, if they are not gamma irradiated. There was, for a time, a process of irradiating all kinds of meats. And it's an interesting process of where you take, like, ground beef, and you pass it in front of a gamma source, and you irradiate it. Now, there's no chance of you generating radioactive material within the meat. So it doesn't have that kind of energy. But you do shatter the DNA of everything that's in there, and so no bacteria can grow in it, and it's absolutely, completely, and totally sterile. And that's not a bad thing. It may reduce the vitamin content slightly of different kinds of vegetables and things, but it's probably a useful thing. All the food that goes up to the International Space Station is irradiated, so they don't take any chances like that. There was a food irradiation plant in Peoria, and there may still be. For a time, there was irradiated food, and it was labeled with the little radioactive trefoil, that three-cornered flower, which, if you think about that, people are not going to be keen on seeing that on their food. That, oh, it's got the radiation symbol on it. Maybe not. So they've either changed the labeling or they phased it out, but nobody talks about it. UV light works really well. At DMH, I'm sure at St. Mary's, they've got a little machine that looks a little bit like a Dalek from Doctor Who. If you don't know that, don't worry about it. It's a little robot guy, sort of R2-D2 looking thing that they'll roll into the machine, into the room. They'll close the door and they'll put little barricades in front of it. They flick it on and probably has a timer so you can run out. And it is a UV blasting machine. So it looks like a gigantic disco ball swirling really intense black light. So it'd be kind of cool to stand in front of with your neon clothes, but you'd also be exposing yourself to shortwave UV radiation. Happily, our ozone layer blocks a lot of harmful UV irradiation, but you can see with the effects of UV irradiation by how quickly you may get a suntan or a sunburn. And so consequently, if you're getting sufficient UV radiation to have a sunburn, you've done some stuff like change your DNA, mutate things. You form a thymine dimer, which blocks the DNA from replicating properly. And you can either end up with mutations or you can kill cells. So, there's a limit. Trying to do UV irradiation to people, not such a good thing. All right. So here's that little UV robot guy. A couple other things just to throw out here is that dehydration works because you're removing the water from the material, remove the water from the bacteria by osmosis, exposing living creatures to a real high salt environment, sucks all the water out. And a high sugar environment would do the same thing. So that will work. Drying is doing that. What you're doing is pulling the water out and making the salt concentration go up. But you can also salt stuff. And this is a good way of preserving things. So done properly, dried fruit, dried foods, dried meat, they're pretty well stable. All right, so with chemicals, we can break things down into a similar kind of sterilization versus sanitation type subcategories, in that we've got sterilants, and these are killing everything. Endospores, bacteria, viruses, these are really toxic guys. Ethylene epoxide, glutaraldehyde is a particularly nasty fixative and molecule. So no, you can't use these on any kind of delicate tissues or living systems. So then we've got some ways of sort of parsing out. A disinfectant is reducing the microbial numbers. Primarily, or it is used on non-living items, but it would be toxic to the skin. So obviously, we would probably categorize this as a disinfectant. Oxivir TB, who knows? Things like bleach, you can get bleach on your hands, but it ain't great. Hydrogen peroxide, again, you could use this, but it's not something that's the most gentle on your skin. Antiseptics are the next level in that they are reducing microbial numbers, but we can then use them on skin. They're toxic if they're taken internally, though, so don't do that. And then we've got some things that you've heard of, like isopropanol, rubbing alcohol. When they get ready to give you a shot, they put the little pad on your arm. What they're doing is locally sterilizing this area of your arm. Bactine, if you have a little scraped boo-boo. And so these are chemicals that will knock back bacteria pretty much in a generic way. I mean, alcohol is going to be pulling water out and dehydrating things and also causing the membranes to blow apart. Antibiotics are a very specific kind of chemical interaction that is going to be affecting bacterial growth. They can be taken internally because they're not going to affect you. They generally are produced by other microbes, either bacteria or fungi, as kind of a way of secreting out into the soil to limit the number of potential competitors. And just as a little point, people are talking about the concern over taking the vaccine. People take antibiotics all the time, and side effects from antibiotics can be quite severe, and yet we prescribe them and take them without even thinking about it. But there you go. So, another one of the terms we use is a way of measuring the relative toxicity of stuff. And there are a lot of ways you can do this. In the pharmacology biz, we do something called the LD50, which is the dose required to kill off 50% of your test population. And obviously, that'd be too much to take. We don't want to even go close to the LD50. With bacteria stuff, we are talking about something that often is something that is referred to as the phenol coefficient. And this is the killing ability relative to phenol, which is like a really toxic, nasty killing agent. So phenol, there's a small amount of phenol in Listerine. It's what gives Listerine that sort of interesting sniff to it, particularly like old-fashioned yellow, old school Listerine has got phenol in it. And Dr. Lister developed observed that phenol has the ability to reduce the bacteria, and that at low concentrations is actually reasonably safe to put on your skin. Straight phenol will give you really nasty chemical burns. So you got the phenol coefficient. And what you do is you expose bacteria to dilutions of phenol or the test chemical for 5 to 10 minutes, or 5 or 10 minutes, and then you put them onto a plate and you let them grow. So you expose them for just a small period of time, and then you see how many did you kill. And you have a whole series of dilutions, because if you remember when we were doing our plates 2 weeks ago, if you just take a loop full of bacteria, there's a ton. But if you dilute it out a bit, you can actually get where you can count them. Well, in this case, what we're doing is looking for the dilution at which we have dead bacteria and we don't see any growth. And by comparing that, we can make a relative kind of efficacy for how efficient a new chemical might be at killing stuff. So the lowest concentration that shows no growth at 10 minutes, not at 5 minutes, and then to compare that to phenol, the lowest no growth at 10 minutes and not at 5 minutes, and that gives you the coefficient. So what we can say is, if it's less than 1, it's less toxic. If it's greater than 1, it's more toxic than phenol. And so phenol is pretty damn good, but there's definitely stuff that's nastier than phenol. I dare say if it's got a phenol coefficient of above 1, probably stay away from that. All right, so the mode of action for a lot of these things is often fairly similar. So you perhaps know of some of these things. Bleach and chlorine tablets. Again, if you're camping, you might use iodine tablets or iodine solution. Is sort of old-fashioned antiseptic for skin treatments. And generally what they're doing is denaturing proteins. So things like phenol and derivatives, things like the hexachlorophene, which is bisphenol, you can find that. Triclosan, which is used in a lot of things like Physohex and Physoderm, antibacterial soaps and stuff. Thimerosal used to be used a lot in solutions like contact lens soaking solution. Thimerosal used to be used a lot as a preservative in vaccines. It contains a small amount of mercury, small enough that it's probably not doing any kind of damage, but the concern is legitimate, and so thimerosal is generally not used as a preservative in stuff that is either going into or onto people. Silver nitrate, again, sort of an old-fashioned killing agent or antiseptic, in that you can dab silver nitrate into wounds. Little babies, right after they're born, have silver nitrate drops drop into their eye to block bacteria from sexually transmitted diseases they might have picked up during birth. Mercurochrome, again, a very old-fashioned kind of disinfectant. So we can disrupt proteins. Another thing that we can do that will kill bugs dead is disrupt the membrane. And this is where washing your hands comes into play. Soap is a really good emulsifier. Soap is very good at bursting apart the plasma membrane. You remember when we started talking about like making mayonnaise, you're taking the phospholipid bilayer, and with soap, you're disrupting the phospholipid bilayer, and that's going to lyse cells. So if you've got a big cell wall, the cell wall is going to keep things in, more or less intact. But soap is going to dissolve the plasma membrane, and so the cell's still going to be probably rendered reasonably ineffective. Alcohol is also affecting the permeability of cells as well. So soaps, alcohols, these work pretty well. Again, not something you can take internally, but externally they work pretty well. Let's see, other kind of general oxidizing agents. What these are going to do is generate some kind of free radical type material, something that's going to oxidize all these different kinds of groups that are involved in electron transport and stuff like that. So benzoyl peroxide, often used as a topical treatment. Hydrogen peroxide, again, topical. And ethylene oxide is typically, ethylene oxide is a gas, and you use that to gas treat a variety of different things. So this is a sterilant, it works quite well. So, just to introduce us to antibiotics. These are operating at a different kind of level, in that antibiotics are typically carrying out some kind of block in specific activities. So all these previous guys are fairly generic. They're going to work on a wide range of both bacteria and potentially animal cells. So antibiotics now can be a lot more selective. Just as a not complete but partial list of the things that antibiotics might do, inhibition of cell wall synthesis, that's a good way of slowing up the bacteria. Inhibition of protein synthesis. This works because while protein synthesis is very similar, it's not exactly the same between prokaryotes and eukaryotes. So you can block protein synthesis in a bacteria and not have at least too much of an effect on the eukaryotic host. You can also inhibit the nucleic acid operations, either replication of the DNA or the production of RNA. And again, this is based on the fact that while the systems are very similar in prokaryotes and eukaryotes, they're not identical, and so you can inhibit bacterial replication and transcription and not have an effect on the individual. There's also some that will just block various kinds of metabolites and things. Let's go do a little bit more here. So, not that you need to memorize the structure, but we've got some basic classes of antibiotics. And this is some examples of how these things can operate. Penicillin and all of the different kinds of cillins, like ampicillin and amoxicillin, these are blocking the production of peptidoglycan and cell walls. And so, without the production of the cell wall, the bacteria is going to be very sensitive to osmotic shock. It will pop open. And so you have penicillin growing here. This is the mold secreting, or penicillium, secreting the antibiotic penicillin, and you get this zone of inhibition. And ostensibly, this is the way penicillin was in fact first discovered. And the building that we're sitting in, John Leidy was a chemist with, I think, Abbott Labs in Indiana, and he was involved in developing some of the techniques that were required to stabilize penicillin. Erythromycin, you may have heard of. This is blocking protein synthesis, and there are a number of guys that are operating at this spot. That chloramphenicol, which is one that's not typically taken, tetracycline, though, you probably have heard of, erythromycin, they're blocking protein synthesis. Rifampicin is a fairly odd one that is not typically prescribed a lot. It's blocking the generation of RNA. All right. So that's what we have to say about antibiotics and things. We'll talk a little bit more, talking a little bit about gene regulation and how these things get involved, but that's it for now. All righty. So this week's lab, I'll get it out to you either tomorrow or Wednesday morning. I think we're done with microscopes for a while. Whew. We're not going to get into looking at ways of distinguishing different bacteria by looking at the kinds of sugars they can grow on and how they interact with different media types. Primarily, can they survive in them? Can they generate and break them down? And we look to see for gas formation. Can they generate carbon dioxide? And if they're surviving well. And so we'll look at some of these kinds of things. And that's all I know.